Hey everyone, I'm Dr. Fizz, and let's learn some physiology. Again, we're skipping our intro. All right, so this one's probably going to be a pretty short one today. We're going to talk about vision and what that means for us. Well, if you're listening to this video, the last video applied to you. If you're watching this video applies to you. So we're going to talk about how the eye focuses light, the types of photoreceptors, on how they convert light into action potentials, and talk about what it means to have center and surrounds within our light when it comes to photoreceptors. Let's get right into it. All right, so we're going to talk about how the eye focuses light. This really is going to be a short video. I'm pretty sure we're only 42 seconds in. Okay, how the eye focuses light. So if we're looking at this picture right here, let me get my there you go. Let me get my trusty laser pointer out. Um, we want to talk about first how the eye even perceives light. So a lot of us have heard that when you see an image, you're actually seeing the light that's reflecting off of the image due to the different pigments that are in that substance is being bounced off. It reflects um, that specific light that you see. So something blue, I'm looking at my blue water bottle right now. My blue water bottle absorbs all of the light except for blue, just because of the chemicals that are within there. So we see that light bouncing off, and depending on the distance, it gives us a perception about how small or how big that image is, or that object is, I should say. It depends on how far away it is. So that light that's bouncing off of a single point on my blue water bottle has to be able to focus and hit. It's spread out. The light is spread out in different directions, but it has to come back and focus and hit my photoreceptors on my retina in the back of my eye. It's kind of tricky, but what helps is something called the cornea. The cornea will take that light, bend it, and then it will go through this opening as well that'll bend the light even more, and it'll go through the lens of our eye, which will bend the light even more and focus it back, pointing it towards the retina, all on one point. It's super cool. Um, so looking at the basic anatomy of the eye, you have something called an optic disc, and out of that optic disc comes our ganglia cells, which picks up signals, and we'll talk about what signals they pick up from something else called our photoreceptors, our rods and our cones back here. Our cones are different photoreceptors that pick up color, like red, blue, and green. And our rods are what pick up black and white, help us with seeing edges and corners and whatnot. In the back, you have pigment epithelium, which helps us pick up on our blue, red, and green. Talking about two specific things that we need to discuss right now, too. We need to talk about the ciliary bundles as well as the muscles that are surrounding our eye. These ligaments, the ciliary ligaments, and the contractile muscles that are around our eye. So this can represent our pupil and our cornea. Really, it's actually our lens. It's our lens with a bunch of muscles surrounding it. So, in order to focus the eye better, if there's more light, we'll actually see a contraction of these muscles. Sorry, we'll see a relaxation of these muscles, which will cause our eye to kind of turn into this sort of thing. It's actually more of let me draw it. It's actually more of a sliver. We'll see our lens kind of do that if there's a lot of light, so we don't have to let in as much light to hurt our eyes or sort of photoreceptors. If there's not enough light, then our contractile muscles will actually relax, and our ligaments will loosen up and well, actually, hang on. If we see a lot of light, our contractor muscles will relax, causing our ligaments to stretch, which will turn our lens into this. And if we see If we see a lot of light, then our muscles will relax or will tensen up. If we don't see a lot of light, then our muscles here will tense up and contract, causing the ligaments to loosen up, which will open up our pupil and we can see more light. Therefore, so that's what I wanted to say. All right, moving on. We now have blood vessels that line this. Those little, if you ever look at a light for too long and you have little squigglies that are moving around in your eye, those are the blood. Those are the light refracting off the blood vessels, the corners of the blood vessels that are in your eye. It's kind of crazy, and when you look around, the light moves around. So kind of cool. All right, let's move on then. Okay, these are photoreceptors. We have two different types of photoreceptors. One called sorry, one second. Where is that? Okay. We have two different types of photoreceptors, on and off photoreceptors, but we're going to talk about the pathway to seeing light. Basically, light will hit something back here, which will turn our 11-cis molecule. Let's see what it's called. Our 11-cis retinol in this G-coupled protein back here. It'll transform that into 11-trans retinol, which will trigger our alpha protein to move over to PDE. If light hits this, it'll activate PDE, and which PDE breaks down cyclic nucleotides like CAMP, for example. So if CAMP is broken down, that means it won't activate our ligand channels, and no sodium will enter. Therefore, no action potential will occur, like you can see down here. Therefore, no neurotransmitters will be released. If the opposite were to happen, and there were to be no light, PDE wouldn't deactivate CAMP, and that would cause for sodium or calcium channels to be open and allow neurotransmitters out, like glutamate, which is what the neurotransmitter is. That's released from these photoreceptors. It goes into bipolar. So these photoreceptors, or these neurotransmitters, will hit something called mGluR6. mGluR6 is a G protein that's inhibitory. That alpha protein will also go to alpha protein will move to TRPM1, which will deactivate and close TRPM1, which is a sodium channel. If it's a sodium channel, that means it won't depolarize the cell, which means that calcium channels won't open, allowing for neurotransmitters to be released. Therefore, our AMPA, our excitatory receptors for our ions on our ganglionic cells, will not receive signals. Therefore, there will be no opening of channels, and there will be no action potential. Thus, we see dark. So on the bipolar side of it, if there was light, then there'd be no glutamate. This wouldn't be mGluR6 would not be inhibited. Therefore, TRPM1 would be allowed to release calcium or sodium into the channels, which would allow calcium to come in, clip snare proteins attached to the membrane, perform exocytosis, and release glutamate, which would excite our AMPA, allowing for potassium to flow into the cell, creating an action potential down our ganglionic cell. Super cool. Moving on. Okay, so talking about center and surrounds. So every photoreceptor is the exact same. It just depends on what is happening. Generally, every receptor is the same. What makes it different is that in between every receptor, you have a group of them, but generally you have a center and you have a surround. So surrounds are the receptors around the middle of that group, and in between each of them you have a middle channel that causes the release of GABA, which is an inhibitory protein, or inhibitory molecule. And we'll talk about that in just a second. So say, for example, you have your center, and there's light that shines down on it, right? But there's no light that shines down on this guy, nor this guy. Well, you wouldn't release glutamate, right? Or you're releasing very, very little amounts of glutamate here, which would allow for our our cells to send out signals and create action potentials. But if, for example, we had, or for example, we were to not have light here, and it were to be dark all across the board, then we would have action potentials being released here. Glutamate would actually excite the AMPA receptors on these middle proteins, which would release GABA, which comes over to our center, hits something that's called a chlorine channel, and chlorine would flow into the cell, hyperpolarizing the cell and preventing glutamate to be released. That's basically all it is. And if less glutamate were to be released, then that would allow for action potentials to happen here. But combined, this would, if there was light here and no light here, these would both be added into the total receptor activity on this bipolar molecule or bipolar enzyme or protein here. These receptors are set up kind of like this. They're like a triangle period with the ganglionic cell at the end. So these two would be coupled together if there was light on this guy and no light on this guy or this guy. But if there was light on this guy and light on this guy, then these two would be the ones that would be contributing to the action potential. Anyways, I think that's it. So wish me luck on the exam, and stay fizzy.